because the program that I use only allows for 15 minutes of recording, I have to do part two. So the first separation technique that I want to go over is paper chromatography. Chromatography means the separation of a liquid mixture. Okay, so we are talking about liquids here, not solids, not gases. So chromatography, liquid mixtures. Um, there's a piece of paper, you put it in a chamber, and what happens is that you have a solvent. A solvent is some liquid or gas that moves that moves another substance. So for example, here is the start line. You're going to put solvent into this this region of the paper. The solvent is called the mobile phase. The reason why it's called the mobile phase is because the solvent is what moves the individual chemicals upward. So, the first thing you do is you put the solvent on the start line, so it's all over here. Then you're going to start putting samples one by one on each side of this paper. So, for example, on this example of a paper chromatography, on spot one, they placed a sample of caffeine, and the caffeine traveled 7.5 centimeters. On the second one, they put paracetamol, which is a painkiller, um, also known as acetaminophen, which is found in Tylenol and Advil. It's the same thing. So they put this here and it traveled 3.1 centimeters. Then they tested four different horse urine samples because they wanted to know if the horses were exposed to drugs before the race. So they used TLC to confirm this. Um, Once they put the spot in, the solvent caused the chemicals to travel in a certain direction. Different chemicals travel at different speeds. A specific chemical will travel at a specific length. So we have our known values of caffeine and paracetamol, and we have urine samples that have various chemicals in it. So they're gonna separate, and the way you read this is this. Um, so if if anything matches with paracetamol, that means that horse will have paracetamol. So paracetamol is spot two, and it looks like horse C, okay, has paracetamol. Okay, it is the only horse that has paracetamol. And then if you look at the caffeine, no horse has the caffeine. And these other horses, horse 3 and horse 6, or horse A and horse D, have something in common here, but we don't know what it is. It might be something in their diet. I don't know. Um, so just know that paper chromatography is a separation of liquids. It is used in forensics. You can find if blood or urine has drugs in it. You could also separate dyes to see what color the dyes came from. Um, it's very common in ink too. You want to see how much ink there is. You want to see what the ink is made out of. Um, so um, that is what paper chromatography is. Um, okay, so what can you conclude from the results of the paper chromatography trial? We could conclude that um, we could conclude that horse C has paracetamol, that no horse was exposed to caffeine. Um, we could conclude that the liquids were separated, um, and so on. Okay. Paper chromatography just tells us that different chemicals and compounds will travel at different rates. Okay. Now the next, come on, come on, come on. the next one I want to talk about is called column chromatography, and this is also the separation of liquids, um, but it's more of a large scale thing because the the way this works is you have something called silica gel or alumina, and that's placed in um, it's placed on an apparatus that separates the liquids. So here's the apparatus that separates the liquids. Um, it's what do you call it? A burette. Usually, what you use is something called burette. You add silica gel on the container, and what that does is that it attracts different liquids due to polarity. You put a mixture inside in the vertical glass column. It doesn't have to be a burette; it could be any glass object. Um, and the different columns form because one compound is more polar than the other. So as you start adding water or some sort of liquid that pushes the layers down, you're gonna notice that it starts to separate in the presence of this silica gel. If you don't have the silica gel, you're not gonna form layers. So you need the silica gel because you need these layers be um, to be attracted to the silica gel or the alumina. Um, and then from there, you just keep adding water and you separate one liquid at a time. So here, all the yellow liquid, we're gonna separate it, and then once the yellow's gone, we get a new beaker or a new flask, and we separate the red. And then once you have the red, you take out the blue, and then whatever is left over is just what you eluted with. So um, so it says here blue can be eluted with IPA and water. I have no idea what IPA means, but it's probably some solvent. And water is just water. Okay, so two techniques paper chromatography, separation of spots, um, dyes, drugs. Column chromatography is the separation of liquids on a large scale. You add solvent and you elute each layer that's separated because of the silica gel or alumina. The third technique I want to talk about is called filtration. Filtration is really simple. You have a mixture, and the mixture is composed of a solid and a liquid. So, separation using a filter results in residue and a filtrate. So, the residue is the stuff that gets stuck onto the filter. Usually, it's crystals, some wet powder, um, and then the filtrate is whatever goes through the filter. So, that's usually the liquid piece of the mixture. However, sometimes you want this residue to be dry, and that's when you want to form crystals. You know, the crystals need to be dry. So instead of using simple filtration, we use something called vacuum filtration. So filtration is when we want to form crystals, and you add the solid here. The solid gets stuck, and then the rest of the liquid is sucked up by a vacuum. It sucks up air and it causes the crystals to dry, and it causes the liquid be to form here at the bottom. So this would be the filtrate, and this would be the residue, which would be the crystals. Separating by acid. So there's also another way to filter stuff, and one way to do it is by adding acid. So if you add acid to, um, let's say, something that's some other substance, it could result in salts and water. And if you have salts, then you could separate the salts by filtration, by a filter, and water will be the filtrate. Another thing you could do is filter based on density using a pipette. So let's say you have let's say you have two different layers here. This is a green layer, and let's say there was another layer that was blue here. You could separate them just by using a pipette. So just remove all the blue liquid, and then that's one way to do it too in the lab if we have layers. And that would also be considered filtration. Um, Another way is also to use a magnet. So anytime you have um, iron pellets, anytime you have iron or lead or mostly iron is um, what scientists play with. If there's iron, you could separate it by using a magnet. So you don't have to go through all of these objects. You could just use a magnet, you could just use an acid, you could just separate using a simple pipette. You don't have to use these fancy techniques all the time. However, let's say that this mixture is made up of a liquid and a solid, but the solid is so tiny that it's so difficult for the filter to separate it. You cannot use a filter anymore. You have to use something called centrifugation. So in this liquid, there is a liquid, but there's also little particles, and you can't see it. It looks like it's all liquid, but it's not. There's also little individual particles in it. So how do you separate those? You use something called centrifugation. You put the sample inside this device. It's called a centrifuge, and what happens is that it starts to spin really fast, and then at the very end, you're gonna form the liquid, which is called the centrifugate. You don't have to know that, but you do have to know what a precipitate is. So when you spin, it starts to form a solid, and this solid, this pellet right here, is called a precipitate. Um, so that is one way to separate a little sample of one thing from its liquid source. Centrifugation is a process of separation of insoluble materials. So again, we cannot see it, but inside of these test tubes, there are particles that are not dissolved in the liquid. They're insoluble, they're not dissolved, but we cannot see it. So the way we see it is by using the centrifuge. The centrifuge is also known as centrifugation. The tube rotates fast, the denser insoluble particles separate from the liquid, forming a precipitate. The last one I want to go over is called simple distillation. It's very simple. Um, this is to separate a mixture of of liquids. Okay, two miscible liquids, two liquids that are actually mixed together. Okay, they're miscible. They mix together. They form one thing. So what you do is you boil them. You boil the liquids. So here is an apparatus. I believe it's heated up here. You have a sample here. Because every single individual liquid has a different boiling point, the one with the smaller boiling point will boil first, and the vapor will form, and then the condenser will cause the vapor to turn into a liquid, and it'll go on to the other side. So let's say you have salt water. You're gonna boil all the water here, and the water is gonna be collected here, and then the salt will remain here. So that's how you separate salt water, for example. However, sometimes you have a liquid that has many different liquids in it, more than two, and that's when you use something called fractional distillation. There's another device here that you put here. It's called I forgot what it's called, but it's you don't need to know this. There are different names for it. I just can't remember it. It basically has different tubings here, and um, it will separate different liquids into different regions of this condenser. So that's how fractional distillation will work. It separates more than two miscible liquids, while simple distillation just separates two things. Okay.